So 70 years ago, Watson and Crick first discovered the structure of the DNA. And in less than a century, we have gone from uncovering the very basic building blocks of life to actually understanding them well enough so that we will be able to be architects of our own biology. Jennifer Doudna once said, "The more we know, the more we realize there is to know." And that is how the science world has advanced, at an astonishing pace, and as residents of this fast-paced world. I believe that we have to ask the question, so what is next? In 2012, Jennifer Doudna and Emmanuel Charpentier discovered or has changed the entire world by publishing their findings on CRISPR-Cas9, an unprecedented genetic engineering technique that allows for fast and precise edits. That it was, in the scientific perspective, revolutionary. And this scientific technique has allowed us, not just from simply editing the plants or animals, but actually being able to make those edits on human cells and human genes alike. These kind of edits, or the genetic engineering techniques, as we so called, is revolutionary, and will be able to help us with all of the different scientific effects that we are going on right now. But as such, of all of these scientific advancements that we have of today, we have also engineered as well as garnered many ethical as well as scientific debate. In terms of CRISPR-Cas9, the problem arises first in terms of whether or not it is ethically moral in terms of engineering our own genes to fix it for the better. Of course, at the same time, it has opened many new doors. Immunity to deadly and sometimes fatal diseases such as HIV, alleviating the symptoms of deadly genetic disorders such as sickle cell anemia or muscle dystrophia. And it has also allowed us and will allow us to live in a world where astronauts are physiologically safe, protected from space radiation. And also, we'll have literal Captain Americas protecting world peace. And all of this is not a far-fetched story. Already, GMO tomatoes are sneaking into our spaghettis, and the CRISPR-Cas9 system will allow us to consume more refined techniques of genetic modified foods. We'll be able to have wheat with less gluten levels and lettuces that will be able to grow in warmer climates. And right now with the CRISPR-Cas9, we are able to apply those methods to humans as well. In 2019, Victoria Gray has went through the very first CRISPR-enabled gene therapy for sickle cell anemia, and is now one of the first ones to be cured of the once deadly disease. And in 2022, a 19-month-old baby was saved from a fatal death due to metachromic leukodystrophy, which has a huge fatality rate. But due to the gene therapy, she was able to survive. With all of this combined, this paints a very, very idealistic picture. But at the same time, as I repeated, this also means that there is a severe backlash. I've talked about the possible problems, or mentioned that there will be an ethical debate. And in this case, I like to ask the question of, first, do you guys know about designer babies? Designer babies is a practice of changing or editing the embryonic genes of an embryo, and thereby having the power to predetermine a person's entire life, from their looks, intelligence, as well as their physical prowess. It essentially gives the power to predetermine a person, and therefore playing God. And with all of this, this also leads to an ethical dilemma of whether or not of the rich and poor gap as well. No matter how much this kind of genetic engineering techniques have been made accessible, it is going to be expensive, and it will be something that only the rich will be able to easily access. And in terms of that, it is going to, if the so-called top 1% are going to use this technique to make their child literally the best. The poor will not just be systematically disadvantaged, but also biologically indisposed, meaning that this gap will be something that they will not be able to overcome. Then the question remains in terms of, is it ethical to give a complete control of one's life to another? As I mentioned in terms of the designer babies, the CRISPR-Cas9 has the power to exactly do that, being able to play God despite our obvious limitations. And once you're going to move on to that kind of ethical debate, we have to ask ourselves where we'll be able to draw that line. Where can you make that ethical judgment or that scientific judgment so that this will be able to, so instead of falling into this downward spiral of negatives or that horrific control as well as gene rich and poor gaps and everything that we have talked about, I suggest that we take a more conservative approach. While some might say that we need to go all in terms of saying that the possible advancements that these genetic gene editing techniques will bring to human health is far too grand to be able to ignore. But in terms of that scientific limitations, as well as we still not being able to figure out the exact problems or the limitations of our own DNA, we have to keep in mind that we still have that ethical debate looming over our heads, as well as that physically and as well as scientifically limitations that is going to put us at a standstill. So I suggest that we have a targeted moratorium or legal delay, especially on embryonic gene editing, so that we will be, and especially if we do not have those strict ethical boundaries in place, it is going to make the entire ethical problems as a more murky, as well as a more confusing mess, where people will be only trying to work for their own gain instead of working for the grand total of the humanity. So in terms of that, I like to end on this note that we are still evolving. These 70 years ago, Watson and Crick has discovered the structure of the DNA. And these 70 years ago has been great. We have been able to make this great advancements, and not just coming from AI, more medication to help the people. But we are now at a crossroads of whether or not we'll be able to use the CRISPR-Cas9 as a way to help the people, help fix and remedy and alleviate the genetic symptoms and disorders, or whether or not we will be genetically enshrining a class divide. I would like to state that we are still sailing the unknown scientific waters, and we are still charting the unknown biological frontiers. So ask yourselves the questions, what will we do in the next 7 years? Thank you.